Hello guys, so it's microbiologist 1254. So today I want to teach you something about streaking method. The streaking method of the way you streak sample in an agar, that is in a culture plate. How you inoculate a sample by streaking that sample on an agar plate. So today I'm gonna show you different types of streaking, and also I'm gonna explain to you something about the principle of streaking. Then I'm also gonna teach you the importance and the uses of these streaking methods. So stay tuned, make sure you subscribe to my channel, and let's learn together. So as we can start, we need to understand first of all what is streaking. That is streaking or in agar plate. So we need to define what is streaking. So as a layman language, streaking is a fundamental microbiologist technique that is used to isolate a pure culture. It's a fundamental microbiological technique that is used to isolate a pure culture of a bacteria colony from a mixed culture. And you isolate a pure culture from a mixed of bacteria in a culture. So streaking is just a fundamental microbiological technique that is used to isolate bacteria from a mixed culture. That is streaking. So streaking, as we have understand about definition of streaking, so we need to get to know what is the reason to why you usually do streaking in an agar plate. What are the objectives? You need to understand what are the objectives of streaking. So one of the objectives is just to obtain that pure bacteria in a mixed culture. That is one of the objectives, to obtain a pure culture in a mixed bacterial culture. So the other one is just to obtain a well isolated colony. So to obtain a well isolated colony in a way that you can find a colony that is well isolated for you. It's for you to be easy for you to continue with other tests like the biochemical test, the gram stain, or the drug sensitivity test, that is DST. So that is to obtain a well isolated colony. The third one is just to propagate that bacteria. That is the essence of streaking. You just want to propagate the bacteria. You just want to make sure that that bacteria that is in that sample has grown. It's just you want to propagate. So I can start by explaining to you some of the principle of this streaking method. So as you can see here, I have different plates. So I need to teach you something about the principle of the streaking. So streaking method is based on dilution of the colonies by spreading them. It's just based of if you have inoculated you have placed your sample in this agar plate. That is just streaking helps you to dilute those colonies it just dilute those colonies, enable to get a pure colony. So streaking is just a mechanical way of separating the colonies on a mixed sample, on a mixed culture. So you separate the colonies. That is the one base principle of the streaking. So a sample is just picked. When you have your sample, you just pick your sample by use the wire loop. We have the wire loop here, as I can show you. I have a wire loop here. We have this. We have two types of wire loop. We have this one, the metal one, which is the reusable. You can use it, but we have this plastic one, which we only use it once. So it is disposable, but this one is not disposable. So they have different gauges. As you can see, this loop has a smaller gauge. This, as you can see here, it has a smaller gauge, while this one, it has a bigger gauge. So different loops have different gauges. So mine, I'm going to use you can use either the plastic or the metal one, that is the wire. This one is reusable. So I was talking about the principle of this streaking method. So a sample, you just pick a sample by use the wire loop. So you just pick a sample. For example, we can have a sample like this one. Let's say this is our sample. So you just immerse this wire loop into the sample, the diluted sample, that is. Then you just pick the loopful. When you immerse this one, it will pick a loopful. So you just place it on at one point, the edge of this plate. One point, just place it at one edge of the plate. So after placing it, now that is where streaking starts from. So after placing the sample, you just start streaking. So there are different method of streaking. So I will also going to teach you. So you just pick a sterile wire loop, place over an agar surface. After you have picked your sample, you place over an agar surface, then spread the petri dish to smear the sample. So smear it successful by streaking, that is, you smear it after you've placed your sample here, you smear, then you start streaking, that is, over an agar media in a different patterns. So you just streak on different patterns. So as the streaking continues gradually, the colonies start to isolate itself. You start isolating the colonies at different points. So starting from where it is highly concentrated, as you're streaking, the part that continues gradually, you streak gradually. So in this process, you're spreading the colonies, you're spreading the culture, you're spreading the mixed culture. So you're spreading as you're streaking, you're spreading the pure culture, that is the mixed culture. You're spreading as you're streaking, continuous, just like that, until the last streak, whereby you will get the last streak. It will also help you to get that an isolated colony. So after making those streaks, you just incubate the plate and let the bacteria to grow. So as you can see here, after the last streak, the concentration, it decreases gradually, make what we call a colony forming unit. That is the CFU. The CFU is just a unit of a colony. The colony is just forms a unit. You just obtain a colony that is high, just like a colony like this one. Let me show you. You just obtain a colony that is highly isolated, that is like this one. You can see this one colony, this one, it is isolated. You can see this one is isolated. You can see this one is isolated. You can also see this one is isolated. So that is a colony forming unit, that is the CFU. So after understanding that principle, it's just the principle is just based on dilution of the mixed culture by streaking. After making those streaks, so those streaks dilute the sample and the culture, making it to obtain a colony forming unit. So principle is just based on dilution of the concentrated mixed culture, forming a colony forming unit. So after that principle, now I'm going to show different types of streaking and also explaining it to you the different types and the way we are supposed to form this, to streak these forms of streaking method. So the first method of streaking, we have 4 methods of streaking. Of course, the first method, we have the quadrant streaking method. We have the second one, which is a continuous. It is called continuous streaking. We have the third one, which is zigzag streaking method. Then we have the fourth one, which is called a radiant method, that is radiant streaking method. Then we have the fifth one, which is called the T-streaking, that is T-streaking method. So, and we also have the other one, which is called the semi-quantitative streaking method. Yeah, it's about 6, but semi-quantitative method is just the other one that was formed latest. So I'm going to teach you, I'm going to show you these different types of streaking. So I will start by the first method, which is quadrant method. For quadrant method, I have different plates here that I can show you, showing different type of quadrant method, streaking method, that is. So for quadrant method, streaking is just whereby it is a method whereby a plate, an agar plate, is divided into 4 sectors. It is divided into 4 quadrants. That's why it's called a quadrant method. So the first quadrant, the first sector here, you can see here we have the first sector, we have the second one, then we have another one here, and we have the fourth quadrant here. So these are the sectors. So sector 1, 2, 3, then 4. You can also see this one, 4 sectors. You can see we have one sector, we have another one, we have another one, and we have another one. So those sectors, that's why it is called a quadrant method. So it is simply dividing a plate, an agar plate, into 4 sectors by spreading. You streak the plate into 4 sectors. This method, it is the commonly used method, and it was first used in by this man called Loefflers, and the other man who is called the Grafke. I think they were used, the first guys who were using this method in Robert Koch's laboratory. So they highly practice this type of streaking method, which helped them to isolate these pure colonies. So for our case, this quadrant method, as you have understand that it is divided into 4 quadrants. So the sector which is streaked first, this is the first streaked sector. You can see this one. It has the highly concentrated of the cultures, that is bacteria. Those bacteria are highly concentrated here, as you can see this one. The first sector here, it is highly concentrated. As you streak, they are going to be diluted downward. You can see the second sector, you can see the third sector, and also the fourth sector. So as you're streaking, you're diluting those concentration, so forming a colon forming unit. So I can show you easily method, just a method of streaking, how we are going to do it. So as I had told you earlier, so you just pick your colony, your sample, by using the wire loop. So you just place at one edge of the plate, that is, just place the colony at one edge of the knee, then you start streaking. That is the first quadrant. You streak just like this, then you turn at an angle of 90 degrees, then you pick, you streak continuously again, just like that. Then the fourth you're making the third quadrant. You're not using the sterile loop, so we're using, you pick another loop, so you're going to spread just like the other one at 90 degrees, that one. Then you're going to make the fourth streak, that is, you're using another sterile wire loop, so you're making the fourth quadrant like that. As you can see there, you can see we have quadrants. We have the first one, we have the second one, we have the third one, and the fourth one. So after forming all that, after streaking that, it will appear like this. You can see this was the first streak, then we have the second sector, we have the third sector at this point, then we have the fourth sector at this point. That is a quadrant method. Or you can use this one. You can see using this one, you can see the way sector are divided. So that is the quadrant method. You just divide the plate into 4 sectors. So the second method of streaking, it is called the continuous method. That is the continuous method. It is just I have a sample here. So This is the type of example of a continuous streaking method. And for a continuous streaking method, is just you are distributing the inoculum evenly at a continuous manner. You are distributing the inoculum continuously from one point, from the first point to the last point. That is, you're distributing it evenly. So for this one now, you just pick your sample. You also use this one. So you just place sample at one edge here. Then you make it continuous like this, like that one. It is just a continuous line running from the point of origin. As you're running it continuous, it starts diluting the inoculum, forming it it makes it forming a colony forming unit. As you can see here, I have mine here, which I had done it. It is a continuous. This is the point of origin whereby I started my inoculum. So I just do it continuous. I just do it continuous, forming something like this one. It's just a continuous law. One advantage of this method is that we are not going to use or sterilize the wire loop continuously. We are just using the sterilize only once. You pick the inoculum, you place it to the plate, agar plate, then you just inoculate it continuously. So the other method is the radiant method. I'm going to show you the radiant method. So for the radiant method, the colony, I just place it at one edge point. The colony is just placed at one edge point here, and you can see this one at this point. As you can see at this edge point. Then the lines are made vertically. You make the line vertically. As you can see, the vertical lines are running from the point of origin here. That is from this point, this one, it runs vertically to the radiant, to the diameter of the plate. So after that radiant method, after that vertical lines are made, then the horizontal line starts to streak. You start to streak horizontally to the plate. So this is the second method, the radiant method. You just place it to this point, then you just streak it vertically like this one. Then a radiant, you just make also a vertical streaks, horizontal streak. That is, this is horizontal streak and these are the vertical streak. So you make a horizontal streak like this one, make it to form a colony forming unit. As you can see here, mine has separated very well. As you can see, we have a pure culture isolated. That one, we have that one, we have another one. You can pick any colony at this point because the inoculum has been diluted. You can see here we have the point of origin. It is highly concentrated. As we are streaking, we are diluting the inoculum, so it forms this colony forming unit. That is the CFU. And also we have another method of radiant method. We have this one. So the sample is just placed at the center of the plate, and then you make a vertical streak running to the edge of the plate. That one, horizontal streak, vertical streak, that is, running to the edge of the plate. This is another method of radiant method. So we also have the T-streaking method. The T-streaking method. So the other method that I was showing is called the T-streaking method. So for this one, the plate is divided into 3 sectors. You can see here, it forms a T. We have the first sector, we have the second one, and we have the third one. So it forms this, the first sector, we have the second one, and we have the third one. So here in this one, so the bacteria, the inoculum that is placed at this point. Then you just spread the inoculum like that at that point. Then you do it continuously to the other sector. So you just place, also you run it continuously. You spread like that one, like that one. So you make the third streaking on the same loop. You just pick and spread it on that manner. so it forms something like a T. You can see this one, that is a T sticking method. For T sticking, that is, it forms something like a T. It's just continuous. You just inoculate, stick here, you just stick on the second part here, and just inoculate on the third part one, also forming it something like T. But remember, the plate is divided into 3 sectors, forming it a T like. So on the other method, we have a zigzag method. And a zigzag method is just another type of streaking. That is, it is like it is the same as the continuous method, but this one, the streaks are made in zigzag way. So for this case, we have our primary inoculum here at this point. We just place our inoculum here at that point. This is. Then you make streaks zigzag in zigzag manner, turning from the top to the bottom here. As you're making those zigzag streaks, it is diluting the colonies, the inoculum. So as you can see here, we have an isolated colonies here. We have these colonies that are isolated. So it is easy for you to do any other test with that colony forming unit. So for that one, and also we have the other method, that is the spread method. That is, you just plate, it is used for the liquid inoculums. That is, you just place, you flood the inoculum in this plate, then you just using a spreader. You spread it all over the surface of the agar. So that is the other method that is used in the streak. So that is all